Beleza, vou abrir aqui, peraí. Você passou por e-mail, Magda?
Sim. Não chegou? Não, não saiu daqui.
Eu não recebi nada ainda.
Ai, que inferno. Peraí. Mandei de novo. Está enviando aqui. E nossa internet tá boa.
Não veio também para mim, não.
Tá legal? Tá indo, só que está super devagar a internet. Está indo a passo de tartaruga.
Ainda não chegou.
Não, porque no, ainda não saiu. Fíjate, Javier, por favor, te mandé ahí un email con copia para Igor. Fíjate si podés encaminar, porque a mí me llegó rápido cuando vos me lo mandaste. Fíjate si le podés mandar a Igor el PDF, por favor.
Não, chegou, peraí.
Ah, tá.
É porque são 11 megas, por isso.
Exacto. ¿Consiguió?
Ué?
Agora sim.
Peraí que eu vou tirar meu microfone aqui, tá?
Ahora podés abrir un micrófono, Javier.
Now it's okay?
Sí, ahora sí. Ahí vos
Okay.
pedís para Igor cambiar el slide. Bueno,
Okay.
pero ahora entonces vamos a hacer una rápida presentación. Bueno, disculpen los problemas técnicos. Ya estamos online também no YouTube, né, Leandro? Ok. Então, sejam todos bem-vindos. Hoje temos a Javier Botto, professor da Faculdade de Agronomia da Universidade de Buenos Aires, e ele vai falar um pouquinho sobre fatores de transcrição e o potencial biotecnológico para a obtenção de genótipos superiores para combater as mudanças climáticas. Te agradeço, Javier, a presença. E só para você saber, Javier, si esta presentación queda en el YouTube grabada, si tenés algún dato que sea no publicado, que después quieras que no quede disponible, nos avisas, ¿está?
Sí, no problem. Thank you very much, Magdalena, for your presentation. Sorry, there is return. Well, thank you very much, Magdalena, for presentation. And today I will talk about transcription factors and potential biotechnological benefits to obtain superior genotypes in a global climate change. I will show to you data of my laboratory, because our laboratory are interested to understand how plants integrate environmental information into growth and development. And we are interested in the molecular mechanisms of physiological responses, as well as in the implication for the agriculture. My lab is at Faculty of Agronomy in the University of Buenos Aires. Next, please. Well, phenotypic plasticity is the capacity of a single genotype to exhibit a range of phenotypes in different environments, as you can see in the photo. You have two photos. In one case, you can see that high temperature increase the growth of plants of Arabidopsis thaliana, and in the second photo on the right, you can see Nicotiana tabacum plants that are grow pecíolos and stem when the light environment change and the low red fat ratio decrease for the presence of plant of vicinity of neighborhood plant. The phenotypic plasticity is a consequence of genetic diversity of the genomes, and also because there are different mechanisms and genetic diversity in the signaling mechanisms. And this is the point of our laboratory, focus the attention to understand the regulatory mechanisms of plant response to environmental signals. Next, please. Está activado. ¿No me escuchás, Magdalena?
Sí, te escuchamos perfecto.
Okay. Well, at molecular level in the cells, the phenotypic plasticity is present at different hierarchical levels from the genome, transcriptome, and proteome, that the proteins activate a different phenotype when the plants perceive different environmental signals and endogenous signals that affect the different processes and the biochemical processes in plants. Next, please. In this nice example, you can see three different flowers of three different species of Mimulus. The conceptual signaling processes is, you can see that the phenotype of each plant, of each flower of Mimulus, is the consequence of different patterns of signaling mechanisms. You have three different signaling processes and different networks represented for the same proteins, but the connection between different proteins and the interaction between different proteins, you can have different phenotype. And then this is the idea of our work, to find the signaling, to decipher the signaling network that respond the plants to different environmental signals. Next, please. Transcription factors are one of the proteins very important to decipher different mechanisms in plants. In Arabidopsis, for example, the genome encodes 1.8 thousand different transcription factors grouped in different structure group and different type of B-box of transcription factor. And one of these transcription factor is the B-box proteins. Please, the next. The B-box proteins is a class of transcription factor represented in plant for 30, 40 gene difference that all share the same structure. The B-box transcription factors are thin fingers because the protein are stabilized for thin ions, and they control the signaling network of plant by protein-protein interaction and also by the protein and DNA interaction on the promoter of the genes. The different B-box proteins are classified because for the presence of one or two B-box domain in the amino region responsible for the protein interaction, and for the presence of the CCT domain in the carboxy terminate of the protein responsible for the transport of the protein between nucleus and cytoplasm. The next, please. Well, when the B-box transcription factors share a high homology between different species in plant because they are highly conserved during evolution. In this study, we can study the homology of 214 B-box protein belong to 12 different species, and all are grouped in 5 structure group. The structure group of B-box protein depends if there are one or two B-box or the presence of CCT domain. In our laboratory, we focus in the function of member of B-box protein group 4 and group 5. The difference of structure group 4 and 5 is the presence of one or two B-box, as you can see in the slide. The next, please. Well, in this talk, I will show some result of B-box 24 in seed germination, and also the function of B-box member of structure group 4 and 5, B-box 21, 24, and 28 in shade avoidance responses. And also, I will show some results about the function in the photoinhibition and in the drug tolerance of potato plants that overexpress the BBX21. Next, please. Well, the study of the function of BBX24 in seed germination was belonged to the study of one of my students, Tai Chiorito, and she found that the mutant of BBX24 germinate similarly than wild-type Columbia seeds. However, when we imbibed the seed in moderated saline and osmotic conditions, the mutant of BBH24 germinate worse than wild-type seeds. That means that BBH24 promotes the tolerance to stress conditions. Pharmacological experiments with different concentration of ABA and gibberellins in the medium where the seeds germinate demonstrate that BBH24 mutants are more sensitive to ABA, and also the germination response to different concentration of gibberellin was affected when the BBH24 is absent. That means that BBH24 increase the salt and osmotic tolerance of the seed to promote the germination in these conditions. And the role of BBH24 is the inhibition of some ABA signaling genes like AB3 and AB5. You can see in this figure that the mutant of BBH24 have more expression of these two signaling ABA genes. Next, please. In another study done by Carlos Croco, that he was a PhD student of my lab, he focused his PhD, he studies in the role of BBOX transcription factor in shade avoidance responses. The shade avoidance responses is a syndrome of plants to compete for light for photosynthesis, and the plant promote the growth of petioles and stem to compete for light. The idea of this work was to study the function of the BBOX members of structure group 4 that I remember, that is the BBOX transcription factor encoded with 2 BBOX domains. In the figure, we study the phenotype of BBX member mutants, cultivating the seedlings under light filter that reduce the red photon, simulated the shade condition when plants grow together. You can see that some members of BBX, like BBX18 and BBX24, promote the growth in these conditions, and BBX21 and BBX22 inhibit the growth. Then we focus our attention about the mechanisms that control BBX21 in shade avoidance response, and we confirm by different alleles that BBX21 inhibit the growth under shade. The expression of BBX21 in wild-type seedlings under shade is promoted rapidly after 1 hour of shade signals. Next, please. To find out the gene regulated by BBX21, we did a microarray analysis, and we found that more than 500 genes are regulated by BBX21 under shade. Approximately 200 of those genes were downregulated by BBX21 that repress the growth under shade conditions. These genes are enriched in auxin, brasinosteroid, and ethylene signaling members. Next, please. In addition, we confirmed the expression of PAR genes in shade avoidance. The PAR genes are responsible for inhibit the the activity of PIT4. PIT4 is one important transcription factor that promotes the Shay avoidance response, and the PAR genes are inhibiting the response of PIT4 to avoid exaggerated responses to Shay. In this case, on the left, you can see that 4 PAR genes are upregulated by BBH21 because the mutant significantly express low level of these genes. In addition, BBH21 can be degraded by COP1 that belong to the proteasome system of the cell. And also, COP1 is known to promote the Shay avoidance response. Then we ask about the genetic interaction between BBH21 and COP1 in Shay avoidance response. By genetic analysis, we demonstrate that that BBH21 and BBH22 operate in the COP1 signaling. As you can see in the figure, the mutant of COP1 did not respond to Shay avoidance signal, and the double mutant of BBH21 or BBH22 with COP1 also did not show Shay avoidance response. However, the triple mutants of BBH21, 22, and COP1 rescue partially the Shay avoidance response in seedlings of Arabidopsis. That means that BBH21 operate in the COP1 signaling to induce the Shay avoidance response, probably regulating by a feedback negative loop the PAR genes to avoid exaggerated response to Shay avoidance. Next, please. As you remember, in the Affymetric analysis of BBH21 regulated gene under chain, we detect that brasinosteroid genes are overrepresented for those genes regulated by BBH21 under chain. To have a more precise idea about the action of BBH21 in brasinosteroid signaling networks, we expose the seedlings to different doses of brasinolide. This is a synthetic hormone of brasinosteroid, and we found that the mutant of BBH21 did not respond to brasinosteroid. In addition, another pharmacological analysis with brasinolide demonstrate that BBH21 mutants are more sensitive to brasinosteroid than wild type under shade. And one of the mechanisms that we discovered in this paper was that BBH21 regulate the expression of genes that induce active brasinosteroid in the plants. In particular, we found that the degradator BAS1, that is a gene responsible for the degradation of brasinosteroid in the tissue, are upregulated by BBH21 under shade. Next, please. In addition, we studied the BBH21 regulated gene and co-regulated by another signaling branches of PIT4 and brasinolide resistant one, that is a transcription factor that inhibits the brasinosteroid response. We studied which we found out the gene co-regulated by the three signaling networks, and we found that in some co-regulated genes are downregulated by BBH21 quickly when the seedlings are exposed to shade. Only with only 1 hour of far red, some members of gene are downregulated by BBX21, like transcription factor MIB-like-2, or PREG1, or PREG3, or IEE4. All these genes are upregulated when BBX21 is present in shade. Next, please. Well, in addition to BBX21, that is a B-box with 2 B-box domain, BBX24 is another member of a structure group form of B-box transcription factor. And in a previous work, we found that BBX24 has opposite role in the shade, promoting the growth, and the role is opposite to BBX21. The idea is that BBX21 inhibits the growth and BBX24 promotes the growth under shade. The idea of this work, published in Nature Communications, was to find the mechanism regulated by BBX24 under shade. We confirmed by 2 different alleles of BBX24, that BBX24 protein promote the growth, while the overexpression lines are taller than wild type under shade. The BBX24 interact with PID4, promoting the growth of seedlings. Next, under shade. Next, please. By an analysis of the transcriptome to find out the gene regulated by BBX24 under shade, we found an enrichment of gene expressed with cis domain represented by member of auxin and gibberellin signaling elements. By a pharmacological approach, we found that gibberellin rescue the phenotype of BBX24 mutants, and the BBX24 mutant were more sensitive to gibberellin than wild type, and the opposite phenotype was observed for the transgenic lines. Next, please. Well, one potent inhibitor of gibberellin signal is DELA. The DELA is inhibit the gibberellin response, and we asked about the possibility that BBX24 interact with DELA. By S-Shook hybrid, we demonstrate that BBX24 effectively interact with DELA, and also we confirm the results by different techniques in vivo, like bifluorescence complementation and co-IP. In all cases, we confirm that BBX24 interact with DELA. Next, please. Well, previously to our work, it has been demonstrated that PID4 can also interact with DELA. And also, we know that PID4 is an activator of shade avoidance response, modulating the exchange expression of shade-induced genes. Then we asked about the possibility that BBX24 sequester DELA to allow that PID4 activity increase its expression. In a transient experiment in Nicotiana cells, we demonstrate by a PIL1 reporter genes that Effectively, PID4 promote the PIL1 expression, and the DELA repress the PIL1 expression in a presence of PID4. If our hypothesis is correct, we expect that the presence of BBH24 sequestered DELA and then allow that PID4 expression promote PIL1. Effectively, in this expression analysis, we found that in the presence of PID4, BBH24, and DELA, the phenotype that PIL1 expression is increased. And that means that our model is correct, that BBH24 sequester DELA in shade and allow PID4 promote the gene expression associated with the growth that allow plants grow in shade. Next, please. Well, another line of work in our laboratory was study the function of BBH28 protein in shade avoidance response. BBH28 protein is a member of BBOX, belong to the structure group 5 that has only one BBOX. Maite Saura did her PhD studies about BBOX in shade avoidance, and she found, as you can see in the photo, that the mutants of BBOX 28 has inhibited the hypocotyl growth under shade, and the overexpression lines of BBX28 promote the growth under shade. The experiment was done using filters that simulate the shade light. The shade light reduces the red photons, the blue photon, and also the irradiation that reach the plants. That means that different signals of light induce the shade avoidance response. The question that we want to respond to answer in this investigation was which signal of shade is responsible. What happened? Which signal of shade was responsible for the activity of BBX28? In the figure below the photo, you can see that the shade signal responsible for the shade avoidance response associated with BBX28 is the changes in the red-far red ratio, because the reduction of blue or the reduction of light intensity did not show differential response between wild type and mutant plants. Another question was how the BBX, which is the mechanism of BBX28 action in the shade. In the figures on the right, you can see the growth rate of seedlings during the first day of shade. The wild type, you can see that during the first photoperiod of exposition of seedling under shade, show that the shade signal increased the growth rate during all the photoperiod. However, the mutant seedlings of BBH28 show impaired response at the end of the photoperiod of shade, and while the overexpression lines maintain the growth rate at the end of the photoperiod. That means that BBX28 has activity to promote the growth at the end of the photoperiod of the shade. Next, please. Well, we analyzed the expression of BBX28 under shade, and we didn't found difference explained by the gene expression of BBX28. And then we asked about the stability of BBX28 protein using reported green fluorescence protein reporter gene associated with the expression of BBX28. You can see that BBX28 in confocal image is stabilized by the end of the photoperiod, because the BBX signal is higher during all the photoperiod compared to wild type. In addition, immunoblot analysis show that in simulated shade at the end of the photoperiod, after 6 hours of shade, the BBX28 protein is more stable in shade than in white light conditions. Next, please. Well, we want to find out the BBX28 regulated gene in shade, and we found that approximately 700 genes are upregulated by BBX28 in shade. Between those genes, they are enriched in auxin transport, auxin biosynthesis, and auxin signaling genes, and also cell wall growth and cell site regulation. All of these ontology groups are associated with the activity of BBX28 promoted gene that facilitate the growth of the cell through auxin signaling. We confirm the result of the transcriptome using some evaluating the gene expression of auxin signaling and auxin transport genes. You can see that shade affects significantly the expression of auxin gene in wild type, while the mutant of BBX28 has impaired the response to promote the expression or to regulate the expression of these genes. This result confirms that one mechanism of BBX28 action is through the auxin signaling network. Next, please. Well, as BBX21, BBX28 also interact with COP in darkness. COP1 degrades BBX28 in darkness. It was demonstrated by the group of Xu some times ago. Then, because COP1 also regulates the shade avoidance responses, we asked about the possibility that the stability of BBX28 at the end of the photoperiod of shade is mediated by COP1. In this study, we evaluate the genetic responses of simple and double triple mutants of BBX28 at COP1. As we expected, COP1 mutant did not respond to shade signals because the seedlings are very short. The the double mutant between BBX28 and COP1 also don't respond to shade signals. However, the overexpression of BBX28 promotes significantly the growth response in shade. Interestingly, and the most interesting data of this experiment is that the double mutant of overexpression BBX28 in the COP1 background mutant, the response of shade disappear. That means that BBX28 needs the presence of COP1 for their activity under shade. These genetic analyses were complemented with confocal image demonstrating that the stability of BBX28 depends on the presence of COP1. In the image of confocal on the right of the slide, you can see that in the COP1 mutant, the BBX28 is not present. And in addition, a complementary study using a potent inhibitor of COP1 function demonstrates that COP1 is a prerequisite to the activity of BBX28 under shade. And these confocal analyses were complemented with immunological analysis, where we found that wild-type seedlings show a high expression of BBX28 in shade, but when we use the inhibitor of COP1 proteasome, the presence of the stability of BBX28 disappear. And this result confirms that the mechanism of BBX28 action to promote the growth under shade depends on the presence of COP1 to regulate auxin genes. Next, please. Well, in the last part of the talk, we talk about our result using in potatoes. We want to understand the function and the mechanism of BBX21 overexpression in potato plants. Previously, we can see that Arabidopsis plants that overexpress BBX21 are plants more robust that have more branches, flowering branches. And then we design an experiment to generate transgenic lines that express in potato plants the gene of Arabidopsis of BBX21. We generate 3 transgenic lines that overexpress BBX21. The name of this transgenic plant is CH1, 2, and 3. In the photo, you can see that overexpression plant of BBX21 are shorter than wild type when they grow in a greenhouse. In addition, the overexpression plant has wider stems and wider leaf. Next, please. The transgenic plant has also modified the production of tubers. In this photo, you can see that the number of tubers per plant are higher in overexpressing line, and also the consequence of more tuber is that the production of tuber is higher than wild type plants. In addition, when we did cut of the tissue of tuber, we found the number of amyloplasts in overexpression line are lower than wild type, but the size of amyloplast is higher than wild type. Next, please. Well, we also study the photosynthetic behavior of transgenic plants under natural conditions in overexpression plants of potato, and we found that the photosynthesis rate and other parameters of photosynthesis like transpiration, stomatal conductance are also higher in transgenic lines than in wild-type plants. This response is associated because the transgenic plants has the stomata, the opening of the stomata is higher than the wild type. There are no difference in the number of stomata. The difference is that the stomata is bigger. The whole of the stomata is bigger than wild type. And in addition, the chlorophyll synthesis is higher in overexpression plant compared with wild type. And this is also related with the more expression of rubisco-activasa gene that is involved in photosynthesis of the plants. Next, please. In addition to this phenotype of overexpressing plant, we found that overexpressing plant of BBH21 produce more pigments. The plants have synthesized more phenols and also anthocyanins. And this you can see here on the left. And the photo show that the plant, the overexpressing plant that has more pigment absorb more fluorescence than wild type. And you can see this in the photo below. The higher pigmentation of overexpression plants is associated because several genes of the signaling pathway for the synthesis of pigment is increased in the overexpressing lines, and this produces more metabolites associated with the production of pigments. Previously, it has been demonstrated that the pigment control the photoprotection function in plants. Under high irradiation of the midday, the plant uses the pigment to photoprotect the photosynthesis compound to avoid damage. That means we hypothesize that the more pigment of the overexpressing plants in potatoes play a function for photoprotection. For that, we measure the activity of photosystem 2, and we found that when plants are exposed to high radiation, the photoprotection of the photosystem 2 is higher in the overexpressing lines compared with wild type. Under our condition, the activity of photosystem 2 in wild type is produce 25% less photosynthesis or less photoprotection under high radiation, while the overexpression plants only reduce 10%. That means that BBH21 produce more pigments to avoid damage of the photosystems, and this conduces to higher photosynthesis rate. Please, next, Prof. Well, the last result that I want to show is about the high tolerance of BBS20 plants overexpressing 21 under moderated stress of water. This study was done during the doctoral studies of Gabriel Gómez Ocampo that some of you know. Well, in this study, Gabriel exposed the overexpressing lines to a moderated stress during 2 weeks. Here you can see the protocols. We compare the plant's growth under field capacity that represented in green color, and we expose the plant for 2 weeks after 28 days of field capacity. We reduce the irrigation of plant. This reduction during 2 weeks reduce the water potential in the soil, and the water potential in drought condition was 1.2 megapascal, while the well-watered plants show a water potential of 0.4 megapascal. Well, we found that overexpression, that wild type under this stress condition are taller, and the basal leaf show hydric problems. However, the overexpressing line were shorter, and also the leaves will maintain good characteristic, good water potential into the cells. Next, please. Well, in addition, under this stress condition, we found that the wild type produce more anthocyanins, and the overexpressing lines show constitutive high level of anthocyanins. In addition, the wild type reduced the synthesis of chlorophylls, while the overexpressing lines showed constitutive high level of chlorophylls. This higher level of chlorophyll produce more photosynthesis in the plants and better use of water to promote to produce hydrates of carbons. The water use of plant, the water use is higher in all transgenic plants under drought. Next, please. Well, interestingly, a time curve of photosynthesis of this plant demonstrates that BBX21 control the photosynthesis rate during the morning of the day. In this case, in this experiment, we expose wild type and overexpressing line to different drought conditions, and we measure the photosynthesis during all the day. We measure the photosynthesis at 6 a.m., 9 a.m., 12 p.m., 14 p.m., and at the end of the day. The photosynthesis rate of the plant was significantly affected in drought by the presence of BBS21, because the photosynthesis rate was higher, specifically in overexpressing line, when they are cultivated under drug. And this higher photosynthesis in the morning also is showed by a higher mesophyll conductance of dioxide of carbon at 9 a.m. Next, please. Well, we asked about the mechanisms, the photosynthesis mechanisms involved in this response of photosynthesis, and we measure with a Likor 1.400. We measure the photosynthesis efficiency and the ratio of dioxide of carbon in the stomach and in the chloroplast to calculate the ratio of dioxide. In this manner, we evaluate the dioxide of carbon diffusion in the plant tissues. We found that the photosynthesis efficiency is significantly higher between genotypes in drought but not in well-watered condition, and also the dioxide of diffusion into the chloroplast is higher in the overexpressing lines compared with wild type under drought conditions. Next, please. Well, then we observe by complementation analysis that we confirm that the mesophyll conductance of the dioxide into the plant tissues is higher in the overexpressing plant than in wild type under drought. And in addition, we estimate the electron transport capacity of the photosystem of photosynthesis, and we found that the overexpressing line are significantly has electron transport capacity compared with wild type. And these two mechanisms, the increasing mesophyll conductance and the higher electron transport capacity, are responsible for the higher rate photosynthesis of overexpressing plants under drought. Please, the next. Well, we also asked about which signaling pathway will be involved in this response of more tolerant response of overexpressing line. One candidate is that the ABA hormone is responsible under stress condition to tolerate the stress. The plant produce more ABA, and then we study the ABA content in the plant tissue, and in the figure on the left you can see that the ABA content in plant tissues is similar in both conditions in wild type and overexpression lines. However, we found difference in the ABA sensitivity of the plant tissues. We demonstrate by two different experiments, one using the touched leaves and one in enter plants growing in greenhouse, that different doses of ABA induce differences between the genotypes. Here, in the experiment of the touched leaves, you can see that plants overexpressing BBH21 are more tolerant to moderate concentration of ABA, like 1 micromolar of ABA did not produce effect in the stomata aperture of the plants, while in wild type, this ABA concentration produces the close of the stomata. In plants, when we spray with ABA, we found that moderated concentration of ABA induce a more tolerant response for photosynthesis and other photosynthesis parameters, and they are more tolerant to this concentration of ABA than wild type. The hormonal level used was 2 micromolar of ABA, and they, in all cases, the response of overexpressing plant was higher than wild type. That means that BBS21 induced more tolerance to drug through the ABA signaling network. Next, please. Well, we are interested to know the BBS21 regulated sheet under drug, and then we use a transcriptome. We analyze by the transcriptome of gene regulated by BBS21, specifically under shade, and we found approximately two clusters of genes that found that they is differentially regulated by BBS21 under drug. Between these genes, we found that all the genes studied by qPCR analysis were downregulated in wild-type plants, while these genes were downregulated by drug in overexpressing line. Here you can see some, for example, some genes like aquaporins or transcription factor like MIB37 that are constitutive upregulated in overexpression lines, and in other case you can see that some genes like photosynthesis genes like a SHIP1 or slide 3 were constitutive downregulated by drug in overexpression lines. And finally, we found that photosynthesis, stress, and aquaporin are specifically regulated by BBS21 under drug. Next, please. Well, probably you asked about the consequence of the drug in the production of tubers. This was a question that we answered during the experimental approach during the work of Gabriel as PhD student, and we found that the drug affect significantly the tuber weight per plant. Here on the right, you can see that the overexpressing lines produce at least 10% more tuber weight per plant when the plants are exposed for 2 weeks during the vegetative growth. Well, that means that BBS21 has a high potential for biotechnological use. And then next, please, you can see that, well, this result and the technological product was patented for our lab. And now this patent is exploited by biotechnological startups, and to scale this study to other crops like soya and alfalfa. Next, please. Well, I won't wrap up the principal result of this research during the last 7-10 years of my lab. And first, during the research, we demonstrate that BBX proteins with double B box, that belong to the group 4 structure proteins, are involved in the control of seed germination, seedling development. This data I didn't show in this presentation, but was published in a plant cell in 2013, and shareable and response. Then the structure group of BBX protein are very important members to regulate the growth and development of plants. The second conclusion is that BBX protein with a single B box, like BBX28, regulate cell growth responses through BBX28, promoting cell growth in shade light. The third conclusion is that BBX proteins of structure group 4 has opposite function for the regulation of growth response, and they act in different signaling networks. For example, BBX21 was demonstrated to operate in the COP1 signaling and brasinosteroid signaling, regulating the homeostasis of brasinosteroid in the plant tissues, while BBX24 was demonstrated to interact with DELA and sequester DELA, allowing the PID4 activity. In potato, we demonstrate that the overexpression of BBX21 has a high impact as biotechnological to increase the productivity of crops. Because when we grow the plants in natural conditions and optimal conditions, we found that the photosynthesis and the production of tuber were higher in overexpressing plants. In addition, the mechanisms of the regulation of BBX21 for the tolerance to stress, we demonstrate that the higher production of pigments induce a better response for the photoinhibition. That means that BBX21 has a function for photoprotection when plant grow at high irradiance. And finally, hola.
Sí, te escuchamos perfecto.
Because I read that somebody say that the microphone is closed. Well, and finally, we found that the overexpression of BBX21 produce higher photosynthesis in potato cultivated under drought by improving the mesophyll conductance of the oxide and electron transport capacity. Well, finally, the last, please. Finally, I acknowledge all the team of my lab that make possible that our laboratory produce nice science. And here you can see a photo with some member and former member of my lab. In addition, I want to acknowledge some collaborators of my lab, like Ezequiel Petricho, Pablo Cerdán, Marcelo Janowski, José Pruneda Paz, and also Shiran Gangapa and Miguel Blasquez and David Alabadid that work in India and Spain. And also I wish to acknowledge the financial of our research for the FONCIT, CONICET, University of Buenos Aires, and also European community. Thank you very much for your attention.
Thank you very much, Javier, for the nice talk and overcoming technical problems. And do we have any question from the meet? Igor?
I can start. Javier, very nice talk, very nice storyline. from your group. I'm wondering about the results that you showed from the overexpression of BBX21 in potato. We can see there are a lot of different phenotypes, especially regarding the production of like the increase in photosynthesis, and then you have like a higher opening area of the stomata, but also an increase in phenolic compounds, anthocyanins, chlorogenic acids. Is this a direct function of BBX21? Because it seems to me that those are very broad results. I mean, you have like, I can understand that these are transcription factors working upstream in these hormone signalings, but do you know whether this increase in phenolics or this is a direct function of BBX or even BBX proteins, or this might be an indirect result of the overexpression of the, then you have like a downstream network.
We have a lot of data that demonstrate that BBX21 promote expression of the phenols and anthocyanins signaling network. In the slide, you can see that there are some knobs of control of the expression of these pigments in the phenol-propanoid pathway, and in all cases, there are a very good correlation of the presence of BBX21 and the higher expression of this pigment. And in addition, we show a good correlation with the metabolites associated with the downregulation of these genes. And in all cases, the overexpression lines produce a very good correlated pattern of gene expression and metabolites. And this end that the phenotype of the plant produce more pigments. And by luminescence, we show that the pigments synthesis is higher in the overexpression plants.
Okay.
But I think that Igor asked more about if BBX21 directly binds the promoters of the enzyme encoding genes, right? If
yes.
It's a direct. Have you ever tested?
Some previous work demonstrate that the protein of BBX21 can bind to DNA of the promoter of these genes. But in this work, we didn't demonstrate this because it was demonstrated in other species.
I think that Mariana Mariana Mascaro says, Javier, thank you very much for the nice presentation. I wonder whether there is any possibility that the overexpression of BBX could affect the activity of other proteins.
Yes. The answer is yes. There are some results of other laboratory that demonstrate that overexpression of BBX also can affect different function in, for example, in flower plants, affecting the pigmentation of flowers, and also in batata and in apple, there are some evidence that overexpression of BBX changes the production of these plants.
Any other question, maybe in the YouTube? Well, I have one. Actually, Javier, you might know that BBX28, Arabidopsis BBX28, physically interact with high 5. And I wonder whether the observation that you have done about COP1, that COP1 is needed to promote to 28 promote growth is because high 5 interacts with 28.
Yes.
Okay.
Well, the results that document are demonstrated in seedlings, in seedling and photomorphogenesis.
Exactly.
The signaling network of photomorphogenesis is not the same of shade avoidance responses. They are different signaling networks. Some components are common, but not all. Not all. The question is that Y5 is involved in shade avoidance responses? Is probably. I don't know. Some result of Sellaro et al. Of the group of Jorge demonstrate that Y5 is involved in the regulation of shade avoidance response at the end of the day. The most important function regulating the growth is at the end of the day, before the plant go into darkness. And they demonstrate the function of 8Y5 during the sunflex of the shade. That is not the same experimental condition that we show in this talk. The sunflex are a natural condition where the plants are exposed to shade and light during the day for very short time of very short period of time.
Yeah, because of the clouds.
Yes, because of the clouds, because of the leaf movement, because of the wind, etc., etc. And the mechanism, the group of Casal proposed that HY5 is involved for the detection of the sunflux. Yeah, but it's not the same, the experimental condition that we use, because we demonstrate the function of this BBX protein when the shade is constant during all the photo period. That is different, but it's probably that because a lot of BBX interact with Wi-Fi, Hi-Fi, and then it's probably that Hi-Fi could be also regulating the gene expression and the shade when the BBX protein is present or no. But I don't have answer. We need to design a specific experiment, like Casal experiment in the Plant Journal published by Sellaro in 2013, and try to find a phenotype of the BBX28 mutants.
Great. Well, I think that we are run out of time.
But I have one little question.
Okay.
Javier, how conserved is the functions of BBX? Those BBX that you show like characterization in Arabidopsis, then you show like the overexpression in potato, and it worked quite well. Those are eudicot plants. What about grasses? Is the function conserved, or is there any like variation in the function of BBX proteins in monocots, for example?
Well, this is a very nice question. It has two parts. I answer the question in two parts. The first one is that the homology is very high, because in a study that we use 214 BBX belong to mosses, monocot, dicot, etc. Always the study demonstrate that they have a very high homology. The second, yes, and always they classify in 5 structure groups, and this means that they are highly conserved since the primitive times of evolution. The second part is about what happened of which is the function of BBX in monocots. There are not too much papers published with monocots. This is the truth. Most of the work were done in dicots. However, I think that the question is very interesting, and using our patent, we are interested to try it in monocot, the transgenesis of BBH21, because one important effect or one important product of monocot in some cases is the use of grassland for cow, and then you need increase the vegetation of the plant. You need to increase the leaf, and then I think that the monocot is a very nice and interesting crops to try these questions.
Great. Well, thank you very much.
Well, thank you.
We keep in touch. Thank you in behalf of Igor and Leandro.
Yeah, those are very nice.
Collaboration in our discipline. And we had a lot of people in YouTube as well, right?
We have quite some people on YouTube. I was not following because I was changing the slides, but
well, thank you very much for your time, your attention, and your nice interest in our research.
Great, Javier. Great. See you. We are in touch. Thank you very much.
Bye-bye. Thank you.
Bye-bye.
Bye-bye.
Well, thanks, for the students, this is our last seminar. Okay, so thank you very much for your participation. It was a great group this year.
Yeah,
you
indeed.
were really very good participants all along the different talks. So I thank you very, very much to the students. Okay.
Thank you very much, guys.
Well, we are always here. Okay. Thank you. See you. Bye-bye.
Bye.
Bye-bye.